Dr. Jermy worked in several industrial relevance projects, including Saudi Aramco and KFUPM. He holds 12 USA patents and published several articles with H-Index 30. Welcome, Dr. Jermy.
Yeah. Thank you.
You're welcome.
So, am I audible? Am I audible?
Sorry, Doctor?
Am I audible?
Yes, you can proceed. You can start now.
Yeah, okay. Thank you for the introduction. So just at today's seminar, I like to discuss about some of the opportunities of the interdisciplinary field of research between my background is from chemistry, how we can apply such track, okay, for the biomedical application. So what are the opportunities are there and what are the challenges we face? So I will discuss about that. So we'll go to next slide. So just I will discuss about what are the materials we can design for this purpose of biomedical application, techniques, and what are the characterizations we can utilize, what are the biomedical applications we can apply, and what is the gap, okay, between academic and industry type of research, and about the data collection. So this is the overview of my talk. So here you can see that as we have seen, there is tremendous, okay, it's like epidemic, but there are a lot of cancer cases, okay, in the recent years, can be due to several reasons, but we can see that even in Saudi Arabia, okay, so there is the breast cancer, and so what are other type of cancers, there is a sudden increase. So there is so even in the worldwide, there are so many deaths due to cancer, okay, so it's second leading disease, followed by first is heart related. The second is the cancer. So there is we need to have something new. Okay, so there are conventional chemo drugs that has been used worldwide, and it has positive and negative effects. So there is some opportunities, okay, for the nanotechnology-driven based research. So we can see how we can utilize. So as you can see here, okay, so in the conventional classical delivery, we can see that when we inject, okay, the chemo drugs, it pass through the body, okay. So it is not specific, okay, target specific, like it can attack both the cancer cell and normal cell, okay. There is no specificity. So that is the drawback of the chemo drugs. And you can see that through nano medicine, okay, like nanotechnology, we can focus mostly on the cancer cell, tumor region, and we can reduce the side effect. So by this way, we can also able to reduce the dose, okay, and also we are more target oriented. So this is a very good opportunity for us, okay. And there are several nano formulations available in the market, like microgel, nanogel. Okay, so it's on the move. So we can go. So in the next slide, we can see that there are so many leading universities, institutes has already been working on developing the drug delivery system. So what is the advantage? We can see that drug delivery system, it should have very high surface area and pore size, pore volume, where we can engineer or design, we can functionalize like several drugs, like drug A, B, as we can see in the right side, okay, stimuli-responsive polymers, and we can functionalize with antibodies, peptides, and also we can change this drug delivery to multifunctional, like we can load with imaging or contrasting agents. So a lot of scope is there for this developing the drug delivery system. And so in the next slide, we can see that here Okay, so this is the magnetic nanoparticles, which can be used as a multifunctional. It can also act as a drug delivery system. It can also, okay, it can be used for tumor imaging. Okay, how? So there is a magnetic nanoparticle which acts as a core, and we can put, okay, the coating, like a silica coating on the external surface. So because of the silica, there is a vast, okay, surface area available, which can be used for functionalizing several drugs, etc. Okay, like peptides, antibodies, small molecules, even siRNA, DNA. Okay, so this is the multifunctional applications we can go. We can use such type of nanomaterials for hypothermia, okay? Where, for example, using the external magnetic, okay, field, we can direct this nanoparticle to the particular site, and we can even increase the temperature, like 40 degree, okay? So that that particular cell environment, okay, will suffer and die. So like this hypothermia application we can use, and also for tumor imaging we can use, okay? So like this, there are multifunctional applications are there with the nanoparticles. So what is the general requirement, okay, for the drug delivery system? You can see that it should reduce the effect, okay, to the healthy cell, and it should focus on the cancer cell, okay? It should be target specific. And you can see in the right side, the drug delivery. This is the standard profile which is used for a drug delivery system, where we can see that there is immediate spike, okay, burst in the traditional drug release pattern. But in the case of a drug delivery system, it should be in the nominal, like nanocarrier-based drug delivery release pattern. We can see that it should increase initially and then stabilize with the time. Okay, so this is the standard we expect. So what another thing we can consider is there is a pH difference. Okay, in the for example, in the tumor region, the pH is 5.5 to 7.2, 6.6 and 7.2. Okay, so this is the pH which we can utilize our designing our nanoparticles. In normal pH, body condition, the pH is 7.4. So in tumor region, you can see that it is 5.5, 6.6, and 7.2. So we can wrap with some kind of polymers, okay, which can interact at the acidic tumor site and it can open up. Okay, so that is the one thing we can utilize. So focus on the cancer cell. So in the normal condition, it will remain as such. Okay. So like this, these are the opportunities for us. So we can see that here we use a drug delivery study, okay, how it can be done. Like two types are there. One is like dialysis membrane technique. Another one is, okay, we can use the Frank cell type, okay, so it is a permagear. You can see that both are almost similar, but the more accurate one is Frank cell. Okay, it will mimic your body condition. So where we can see that we can perform the drug delivery with time. So in this dialysis membrane technique, we can see that we can purchase the membrane. Okay, so it is also mimic our body condition, the membrane tissue-like. So we can put our developed nanoparticle inside, and with the time we can collect, okay, how from the solution, how much drug released with the time. Okay, so this way we can study this one. So we can go to the next slide. So there are different type of nanoparticles are available. Okay, like polymeric, porous structured silica, carbon-based carriers, and recently the metal organic frameworks, and also there are some like liposomes. Liposome has been used, okay, in the industrial scale, and it has a lot of scope. And also metal organic frameworks also, it can be used, okay. For example, if you take ZIF8, okay, so it is considered, it has a zinc and another one linker, okay. So imidazole linker. So these are all pharmacologically active nanoparticles. So we can utilize like this, okay. So in this type of nanoparticle, we will focus, okay, on the porous structure silica, how we can utilize this material, okay. There are a lot of research also going on carbon-based carrier like carbon nanotubes, okay, mesoporous carbon, graphene oxide, and carbon nanocage like that, okay. And the polymers, the PLGA, dendrimer, hydrogel, cationic polymers, a lot of research is going on. So we'll focus on the structure silica. So as you can see that there are different shapes of pores are there in the structure silica. For example, cylindrical, conical, spherical in portal, interstices, and slits. Okay, so this type of different pore shapes we can utilize that. Okay, to deposit our drug inside. Okay, for the drug delivery system, and it will be unique for each material how it encapsulate and then release. Okay. So, for example, we can see how the hierarchical zeolite, for example, the porous zeolite type, and the monodispersed spherical silica, and cubic-shaped SBS16. Okay, so these three type of nanocarriers, we can see how we can utilize in this cancer, blastocytis, diabetes, and antioxidant carriers. Okay, so we'll go to next slide. As you see here, so this is the zeolites. It is basically built with the silica aluminum atom, and it has the secondary building unit, and it leading to the 3D structure. Okay, this type of nanoparticles has been used for heavily for the water decontaminations. Okay, like heavy metal absorption, even this Mayan is a long time. The human civilization, okay, so they utilized for a water filtration, okay, this type of material. It's natural one, and it's non-toxic, okay, so, and it's easily, we can synthesize in the laboratory, cost effective, and it's highly stable. So it is a lot of advantages are there. And in the petrochemicals, they utilize this type of zeolites as a catalyst, okay, in Saudi Aramco, like that. So this is already, it has a very good scope for the biomedical application because of this pores, okay. So we'll go to next slide. So here you can see that there are different shaped zeolites are there. For example, modernite, okay, phagosite, beta, ZSM5, that is MFI structure, okay. So their pores, what is the difference? Their pores and structure, okay. Some are micropore, some are mesopores, medium-sized pores, okay. So they are basically built with the silica alumina ratio, as we can say. So here we can tune the silica alumina ratio, okay. For a biomedical application, we want less aluminum, okay, more silica. So like this, we can tune it. So what we can do in our research, we use the more aluminum in the petrochemical application, the less aluminum, the biomedical application, okay, like cancer therapeutics, and even for a potential treatment of COVID-19, like this, we can use it, okay. So we'll see that. So here, so there are the zeolites are a conventional one where we can see that it has a micropore, okay, small type of pores. But this will be suitable for certain applications. So, but for the large biomolecules, okay, so we need the large pores. So what we'll do that, we need to make the pore size, okay, a little bigger. So that's why the hierarchical pores comes into play, where we can see that in the left side, there is conventional micropores. But in the right side, once you make the pores hierarchical, means combination of micro and meso, more access, okay, of the biomolecules can be obtained to the active sites. So the next slide, there are different techniques we use to generate the micro and mesopores, like demethylation, templating, recrystallization, deposition. Okay, so we will utilize this recrystallization process. So we can see in the next slide. So there are two approaches are there, top-down versus bottom-up approach. In the top down, okay, you can see that we take the zeolite and we treat with sodium hydroxide, like CTAP. We can use the large pore templates to make it big size, okay, large pore nanocarrier, okay. In the next bottom-up approach, we can utilize the raw materials like sodium aluminate, TOS, tetrapropyl ammonium bromide, water like this, and then we can develop the material. So this is the bottom-up approach. These two approaches can be effective, okay, to develop the large pore silica. So what we develop here is high silica zeolite. So where we can see that we reduce the aluminum content. Okay, we only use the silica source, Ludax, tetrapropyl ammonium hydroxide, sodium hydroxide water, and it will undergo the hydrothermal heating, sodium hydroxide. Okay, so it will form a micro and mesopore structure. So this is the hierarchical type of silica zeolite, which we will utilize for biomedical applications. We'll go to the next slide. So here you can see that conventional mesoporous silica is in the left side. Okay, it is a big large pore, and here in the right side you can see the micropore zeolite. Okay, it's a big crystals, but the pores are micropore, very small type. But in micro and meso, you can see that it is intermediate. Okay, so it will consider also the meso and micro. Because the micropores, okay, its stability increases, the mesoporous stability. Usually the mesoporous are amorphous type of silica, which is not that much stable inside. For example, if you give inside the body, it is very acidic pH, okay, and also so it should able to stabilize, okay. So that's why this type of materials are advantageous, because it will also have some zeolitic character, which increase the stability of the nanoparticles. So here you can see that this type of silicas, for example, depending on the silica alumina ratios, okay, there are bulk from the, for example, the we want to load, okay, the antioxidant, okay, which is good as a free radical scavengers. So, for example, coenzyme Q10 is a very large type of antioxidants. Okay, it contains 10 isoprene unit, whereas ascorbic acid is small. Okay, so we can see that even this type of nanocarriers, okay, that can easily absorb, okay, the coenzyme Q10s, and it depends on the silica alumina ratio, how we change the silica alumina ratio, it interaction with the coenzyme affected. So like this, we can, it is able to absorb even the larger molecule of antioxidant biomolecules. Okay. So in another example, we used, okay, cisplatin. For example, cisplatin is the chemo drug, which is used for more than 50% of cancer treatment. This FDA-approved drug is very effective for the treatment. Traditionally, they used for chemotherapy, but it also, as I said earlier, non-specific. Okay, it is not specific. It will also affect the normal cells, and it will induce several side effects like nephrotoxicity, neuroinflammations, pain. Okay, so the body couldn't, and also there is resistance, okay, to the cisplatin also has been there. So what we'll do, so we try to functionalize such chemo drug on the hierarchical micro and mesopore zeolites, and we found that during the preparation, okay, there is an effect on the activity of the cisplatin. Like if you synthesize, develop in the presence of DMSO, its activity was not there. Okay, there is no intercellular release we found, and we found later that during the UV visible analysis, we saw that it forms stable, okay, tetrahedrally stable clusters. But when you use the normal saline solution, okay, we found that the cisplatin is able to disperse, okay, on the support and exhibit the selective, okay, targeted anti-cancer activity. Okay, so like this, we can utilize this kind of nanocarrier, okay, not only for cisplatin, the other chemo drugs like like platinum complexes like carboplatin, oxaliplatin, tamoxifen, so many. It's a vast, wide open opportunities are there. So in the next slide, we can see that not only for drug delivery we can apply, okay? So we can also make a composite with magnetic nanoparticles, like for example, sympions. It is the super paramagnetic ion oxide nanoparticles, okay? So during, we can also go on the super paramagnetic, the sympions, and we can wrap with polymers like polyethylene glycol. We can see that it can be easily, it can be influenced by the external magnetic field, okay? So we can attract, we can inject such nanoparticles, and we can remove such nanoparticles using the external magnetic field. So like this, multifunctional applications are there for this type of materials. And we can see that the addition of, okay, the same micro and mesoporous zeolites, we can see that the addition of curcumin, okay, to the cisplatin, we found that it is more, okay, active, and it is able to overcome the drug resistance, okay, of the cisplatin. So we can see that there are several studies we did, like drug delivery study, and also we collaborate with the stem cell department, okay, to study the in vitro studies. Okay, so like this collaboration also will give you very good the research activities. So in the next slide, we can see that. So we tested on the MCF7 cells. Okay, so these are the breast cancer cell and HeLa. Okay, so this is another type of cell. We can see that. So here, different nanoformulation we have to take. Okay, for example, the control, and you are developed a nanoformulation control like curcumin, alone, cisplatin, and even combination of curcumin and cisplatin, and then your nanoformulations. So at different dose, we can see that, okay, how effective is here. The combination of curcumin and cisplatin, we found that it's more toxic, okay, and able to kill the breast cancer cell and HeLa, okay. So like this, so it is a very good synergistic combination we can develop. And another type of silica, okay, I want to discuss this, the monodispersed spherical silica, okay. This is the, like the shape of the nanocarrier, okay, influence the biomedical applications, the anti-cancer activity, for example. Why? Because the shape, okay, the larger the nanoparticle, it will be easily eliminated from the body, okay. So the research shows that from below 100 nanometer, okay, the particles stay inside the body more time. So in particular, the spherical shaped silica is attractive, okay, because it can be easily accepted by the cell, okay. So this one was commercially available, so it was around 80 nanometer. Okay, so it's perfect for our study. So what we can do, we can load, okay, such type of nanocarrier, okay, with the drugs and polymers. We found that the 50 nanometers is difficult, okay, there is agglomeration is there, aggregation, but the 80 to 100 nanometer, there is individual spherical nanoparticles are there. Here, so the zeta potential, okay, so also shows the presence of effective diameter. We can see that it's around 80 nanometer, okay. So here, what we did, we developed the multifunctional applications where we took that monodispersed spherical silica, and we made a composite with the spinel ferrites, okay. For example, nickel, copper, cobalt, manganese, spinel ferrites. And then we can see that in this case, the chitosan was used as a biocompatible polymer, okay, to wrap. But we need to optimize certain condition because, okay, the chitosan, it involves usage of this acetic acid. Okay, it's because of that, when we functionalize with the cisplatin, as you can see that in the method 1, okay, the first method, and then we do chitosan wrapping, it didn't show any anti-cancer activity. Okay, because of that, what we did, we first wrapped with the polymer chitosan, and then finally we added the cisplatin. Okay, and then we wash. And then we can see that there is a active anti-cancer activity is there. Okay, so like this, we have to optimize certain steps to make it more active. Okay, for the nanoformulations. So here also, we can also functionalize with the folic acid. Okay, so this is the receptor highly expressed in the cancer cell. So we can functionalize such type of folic acid and the magnetic silica nanoformulation. We can see that in the carboplatin release. Okay, so different pH we can see, like pH 5.6, neutral pH, and pH 2.9, like that. Okay, and we see how the release is there. We can see that around acidic pH, we are able to release more, and then normal pH and as 2.9, okay, it was very less. So this is the optimum thing we need, okay, to develop a nanoformula. It should be more active to release the drug at the cancer tumor as the condition, okay. So this type of nanoparticles, spherical nanoparticles, okay, are very important for brain tumors. Okay, for example, it has a blood-brain barrier, so it's very, very selective. So we can use in that for this 80 nanometer particle because it's homogeneous, very spherical. Okay, so we also tried okay, using a cerium oxide because it's a free radical scavenger. As we know, then the brain tumors and all this more free radical formation, of course. Okay, so this it's a scavenger. Okay, so like this we can formulate nanoparticles, and we can see the effect. Okay, compared to the nanotubes and the spherical silica, and we see different drug release. Here we can see that how effective. Okay, so the designing of nanoformulation. Okay, it has a very drastic effect on the release parameter of the drug. So we can also do kinetic modelings. Okay, so as you are in biomedical engineering, so this is the opportunity. So myself, I am expertise in the nanoformulation designing. So we work, okay, in collaborations. We can make kinetic models, how the release, okay, of the drugs. We can use various models. My collaborator used like Cosmere, Peppas, first order, second order, Iguchi, like that. Okay. And we can see that where it has the highest regression coefficient, okay, value. So we can determine, okay, so which model it follows, okay, your nanoformulations, okay. And also we can see here, so there are some other, okay, how the drug interact, okay, with the nanocarriers. We can also study such type using a density functional theory, okay, to calculate the binding energy between the cisplatin and the nanocarrier. For example, this type of study we can do, where we can see that first, once you perform the drug delivery study, okay, so then you can see what factors influence the nanocarrier. For example, we take the spherical silica and graphene oxide, okay, we made a composite and we functionalize the drug and we see the delivery. So how it interact, okay, the cisplatin on spherical silica and graphene oxide, okay. As you know, the graphene oxide, there's like a sheet, okay. So we can go on the next slide. So here we can see that the density functional calculations, okay, is from our another collaborators. So if performed, so we can see that in the silica, okay, there is an interaction. It's primarily because of the electrostatic interactions, and the binding energy was around 1.5 eV. So when we go to next slide, so here you see that the graphene oxide, there are two types of interactions we observe. Okay, so where we can see that the first one, okay, it interacts in the top and bottom layers. The another one, we can see that it's like sandwich type, okay, where it can interact. So like this, we can see the interactions ability, okay, of the cisplatin between the different supports. Okay. And here we want to show about the large pore type of silica, for example, SP16. So this is the third one I choose, okay, where we can see that this is the cubic shaped type of silica with the ink pore like ink bottle, you know, right? The ink bottle. So in the neck, it will be small and the bottom will be the big, like ink bottle, okay? So this type of nanomaterials can be easily utilized because of its large pores, okay? So where we can utilize, for example, you can see that we can utilize in similarly the magnet symbionts silica composite formation. So here we can, because of the large pore surface area, we can functionalize with sil lengths, okay, like APTS, and we can wrap with the polyacrylic acid. So so a lot of scope is there, okay, because of this large surface area. And we can see that we can also make such material composite with metal organic frameworks, okay, like graphite. Here we can see that the graphite gives a cisplatin release around is a unique, and then the SP16 alone, and this one, the combination of SP16 graphite, it gives a unique release pattern. Okay, so like this, depending on the composites, we are able to influence the drug release pattern. Okay, and in this case, you can see that you can also analyze the how our nanoparticle able to penetrate to the cancer cell by using a TEM. Okay, for example, section by ultramicrotomy. Okay, we can see that our nanoparticles are really target specific and it can reach inside the cell of the tumor cell. Okay, by comparing the normal TEM image and the drug loaded one. Okay, so we can see that our particle is able to penetrate, okay, to the cancer cell very effectively, okay, and damage the membranes. Similarly, we can use such type of large pore silica, okay, like K86 for the parasitic disease, okay, like blastocytosis. So it's not only unique, we can see that the nano formulation, the drug delivery, it can be applied to the vast applications, not only for cancers, we can use for blastocytes. So like this, we can expand, okay, by just designing the nanoparticles. Here you can see that the presence of graphite, okay, because it has zinc 2 plus and imidazole linker, it acts as an effective, okay, and nanocarrier to exert anti-blastocyte activity. As such, we loaded with antioxidant like resveratrol and curcumin. We can see that as such, the resveratrol, it spikes, okay, the release. But the presence of this nanocarrier, it gives a sustained release, okay? So this is the advantage. And this type of large SP16 can also be used for potential insulin, okay, release for the treatment of diabetic mellitus. We can see that diabetes is it's almost like a pandemic level. There's a lot of drug delivery system has been focused, okay. But here the insulin, okay, it is a protein, it's not stable, it's a very large molecule. So we have to choose a particular nanocarrier which has very large surface pores, okay. For example, here we used SP16 pores where we can see that it has a, you can see the XRD pattern, you can see that there's a X-ray diffraction of insulin crystalline structure. We see that in the orange, and then we can see the blue, and we can see in the green, okay? So this is the insulin encapsulated nanocarrier. So the crystalline structure disappears clearly, okay? Even we can use the surface area analysis. We can see that after we load the insulin, okay, its surface area decreases from 800 to 335. So around 40% insulin occupation occurs, okay? So like this, we can utilize this type of nanomaterials. So here you can see that we can do diffuse reflectance, UV visible spectra, and FTIR, okay, to see the interactions, okay. For example, in the blue, you can see that in the FTA, there is no significant increase, okay? But once you load the insulin, there is a bump is there, okay? So it shows the interaction of the insulin. Similarly, in the diffuse reflectance, you can see that insulin have its own unique, okay, absorbance pattern and the silica, but the combination of insulin and silica, you can see the intermediate. So this shows the, here you can see that the TEM analysis, the wrapping of the polymer, okay? We can wrap with the polyethylene glycol on the three-dimensionalized B16. So we can see that in the first case, it is normal silica alone. It has a very good pore channels, large pore channels. So this is the final one. The B, we can see that after insulin addition and the polymer wrapping, okay, so there's clearly a change in the textures. Okay, it has been wrapped with the polymers. So like this, we can confirm with these techniques. And also we can see the release pattern, okay, at pH 7.4, 6.8, and 1.2, how it has, okay. We can compare with other large pore silicas. We can also perform the in vivo study, okay, like using the animals, okay. So we have the animal house facility where we can collaborate. And we see that it is able to normalize, okay, our nano formulation, able to normalize the diabetes, the blood glucose level. So, and also we can see that, for example, we found that one of the material like lysite, okay, it is a clay-based nanotubes, which can be effectively used to encapsulate the dexamethasone, okay. For a potential, okay, COVID pulmonary treatment. So we can see that because the presence in this nanocarrier on the external surface is siloxane surface, internal is aluminum. It is based on the surface. This unique character able to show that the dexamethasone release at 5.6. Okay, so we observe that unique character with this nano formulation. And you know that the SARS-CoV-2 fusion and the cell interaction occurs pH 5.5. So so at this pH, it is able to release. So that is the uniqueness of this material. Okay, so like this, we have to see different characteristics. Okay, the properties of the nanocarriers, and so we can see that there are some mismatch also. So once I just want to share with the students. So what are the academic and industrial research gaps are there quickly? So we can see that there's always what we do in the academic, okay, it's not replicating in the industries, okay? So there's a lot of research gap is there. So we need to work on the reproducibility, for example, okay? So how we can see, for example, in while working on the nano formulations, we need to consider, okay, the important is this, you have to note it down all the timings, okay? For example, if you prepare the nanomaterial in 3-hour hydrothermal time, it will form very nano material-like, okay? And 20 hours and 48 hours, its particles shape changes totally. So these are the things we have to make it careful during the synthesis. We have to know how much time we have to leave for the hydrothermal synthesis. And always you should repeat the same time, okay? And you are able to replicate. You have to know how much, what are the beaker size I use, what are the resource I use, the quality of the precursors, like this, okay? And also you have to consider the gel composition. So here you can see that we have to consider all the silica source, acid, water, everything in this gel composition. Okay, so then only we can replicate the nanoparticles. Otherwise, when you do in the small scale, its activity will be different. When you change to bulk, it will be changed. Okay. And the precursors, okay? So for example, you take 3 type of material as a starting material, but its particle size is different, like 0.5 micrometer, 2 micrometer, 3 micrometer. The end product will be different, okay? Because you take, even though its chemical composition is same, the crystal size is different, so you will get the variable products. So this you should be careful. And also storing of the nanoparticles is very important because with the time, okay, so there is one effect called the Oswald ripening effect. So you need to, okay, so you need to take care of this also, the storage of the nanoparticles, okay. And you can, and next you can see that even the when we characterize using the transmission electron microscope, there is a particle charging effect, which you need to carefully consider, okay? Then there is nanoparticles like nickel oxide, if it is on the nanocarrier, okay? When you focus on during the transmission electron microscope, it will charge and your material will react, okay? And form a big nanoparticles. So these things, you should not get misled easily, okay? So you have to consider these parameters. And also you can see that you are depending on the starting materials, your product will change, okay? For example, if you take a big nanoparticle and your final product will be a composite, but if you take a small particles, okay, as a starting material, you will get a different product. So another thing is, I want to share, if time is there, I think, yes. Just I will share that how to collect the data. Okay, for example, to conduct the experiment, you need to make a study plan. Okay, 1 month before itself. So once you make the plan, you have to make a PPT. You have to enter everything. Okay, what are the main components, subcomponent, and what are the factors I take. Similarly, for the application part, and you have to make a nanomaterial list. Okay, so what are the list I make, and you have to give a coding. Okay, and then once you get the result, you are able to get the figure, and next you can write it out. So this is the way standard procedure we can make. So I like to conclude that so we have discussed about some of the nanoparticles. Okay, how we can develop an effective drug delivery system, and we can apply for different applications, not only for cancer and even diabetic and COVID treatments like this. Potentially, we can develop. Yeah, and I'd like to thank my collaborators and thank you very much.
So thank you, Dr. Jermy, for this nice presentation and valuable information. So does anyone have a question? You can just raise your hand. So I can unmute you, or if you want, you can write it down in the QA. I think we don't have any question from our audience. So I would like to ask some questions, if you allow me, Doctor.
Yeah.
You have explained that there's a relationship between the agglomeration and the size of the magnetic core, right? So is there any agglomeration after the coating the nanoparticle, the magnetic nanoparticle with the silica?
Yeah, that one. And so
I
there
mean,
might be some, because after you functionalize with the functionalization procedure, okay,
yeah.
we observe slight agglomeration is there. Some modification will be there. Yeah.
Okay. Nada, yes, you are on mute now. You can ask your question.
Okay, hello.
Yes.
Hello, do you hear me?
Yes, we can.
Okay. My question is about the like violets. You said that you mentioned for the shape of the violets can affect the delivery of these drugs. Which is the best shape that of especially for violet, and if there is another analog for violet, which is the best shape that the best delivery for these drugs. There is a study up to now like this, or
Nada, your voice
clear?
is not clear.
Yeah,
Doctor,
yeah.
did you get the question?
I didn't get. I just get a little. So, is it
Okay.
possible you
Can
can
you
explain me?
can you write it, please? Write your question so doctor can answer your question properly.
Okay.
Nada, can you hear me?
Yes, I hear you.
Yes,
Do you
yes,
hear me now?
yes. Your voice is not clear. Can you write your question, please?
Yes, I will just do it.
Okay. Okay, we have a question here from Sarah. Although there are many advanced medical solutions for treating cancer, why do we not see any of them applied in hospitals?
Yeah, it's like it's all in the clinical phase. Some are there already. There are so many drug delivery systems in the market. Okay. But in the coming 10 years, we can see a lot of solutions. Yeah.
Okay, Nada, I do ask about which is the best shape of zeolite that may help to have best delivery of drugs.
It's like it depends on the particular drug's structure. Okay. So what are the functional group it has? It depends on that one. Okay. And also what are the chemical composition of your zeolites. Okay. For example, for our case, the cisplatin, we have the chlorine functional and NH2. The chlorine is able to interact effectively on the silanol. So it depends on the chemical composition of the drug and the carrier.
Okay. Do you have any question? I think we covered all questions. I have just one more question, Doctor. You have mentioned that we use the direct delivery system for insulin deliver the insulin, right? Did you use the same composite magnetic core with the silica?
We didn't use magnetic, because we focus because there is we find still now no application of magnetic based nanoformulation in this diabetic. So we restrict to the silica.
Okay, silica only. Okay. Yes, Nada, you are raising your hand. If you have another question, just please write it down because your voice is not clear. We cannot hear you. Okay, big thanks to our speaker, Dr. Jermy, for sharing valuable insights, and to our engaged audience for your active participation. And we look forward to seeing you at the next webinar. Thank you. Thank you, Doctor.
Thank you. Thank you.